It is now my pleasure, and I'm told by Lauren that he's online, to introduce our morning speaker. And then I will do the SRF story after that. But I'm really excited, and Mike, I hope you can hear me. Can we put Mike up and let him not this is my presentation for after, but if we can put the Zoom up and let Mike share his screen while I'm introducing him. I'm just going to read his introduction. I'm just going to read his bio off of his website, and then I'm going to let him take over. Very much appreciate Dr. Tranfaglia coming and talking today. I'll read you his bio so you can understand why I'm so excited. He is the Medical Director and Chief Science Officer of the FRAXA Research Foundation. Mike is responsible for coordinating the Foundation's research strategy and working with the university and industry scientists to develop new therapeutic agents for Fragile X. FRAXA is the organization for Fragile X, autism and related developmental disorders. Mike graduated from Harvard with a BA in biology and studied medicine at the University of North Carolina at Chapel Hill. After obtaining his MD, he stayed at UNC for psychiatry residency training and then entered private practice in 1991 in Newburyport, Massachusetts. I'm dyslexic, I can't read these big words. Mike has specialized in the treatment of anxiety disorders prior to his son's Fragile X diagnosis in 1992, before he and his wife founded FRAXA in 1994. So we're really hearing from a giant here who has a lot we can learn from, and I have cribbed his work already. So Mike, thanks for being here. It's very weird to see you on the podium that I'm standing at. And I don't know if you have slides or not, or if we're just going to stream you, but over to you, and thank you again for doing this.
Thanks so much, Mike. And yes, I have lots and lots of slides, hopefully not too many. So I'd like to share my screen with all of you. Everybody hear me okay? Yep. So thanks again to all of you for inviting me. I'm Dr. Mike Tranfaglia, and I am the Medical Director and Chief Scientific Officer of FRAXA Research Foundation. And I'd like to talk to you today about community-based drug discovery, which is what we've been doing for a long time, and it sounds like what you guys are doing too. So I'd like to share our experiences from nearly 30 years of doing this. And I hope that this will be an inspirational story, although you may find it to be more of a cautionary tale than anything else. I am an expert in this field by this definition, at least, because I can at least claim to have made every mistake you can possibly make in doing what we do. We've learned a lot of lessons over the years, and I really enjoy having the opportunity to share those lessons with other folks in the rare disease community. FRAXA was founded in 1994 by my wife and I and another parent from Northern Virginia, dedicated to funding biomedical research, and we've funded over $36 million in research grants to date. That's over a very long period of time, so it's really not as much as it sounds like, but we like to think we've been pretty effective considering the amount of money we've had to work with. Most importantly, FRAXA is founded on the principle that Fragile X is a treatable condition. And when we got started, this was actually a little bit controversial, but nowadays I think it's pretty well accepted by pharma and biotech companies, as well as the community at large and the larger rare disease community. Check us out at fraxa.org if you get a chance. We do this for the two most important reasons in the world, and those reasons are named Andy and Laura, our kids. And Andy is now 34 years old. He has a classic full mutation of Fragile X, seizure disorder, all kinds of developmental delay and behavioral problems. Laura actually has the exact same full mutation that Andy has, except she's a female. She's now 32, and she's perfectly normal. She's married, she's a college graduate, has a nice job and a house in the suburbs. So she's relatively unaffected. I won't say completely unaffected, but just by virtue of being female, she's perfectly normal in every way. One of the interesting things about Fragile X is the most common inherited cause of intellectual disability. It affects about 1 in 4,000 boys. About 1 in 4,000 girls have the full mutation, but like I said, a lot of them are not really affected in any significant way. And so we usually say about 1 in 8,000 girls are significantly affected. Most common cause of autism. Fragile X has a fairly subtle physical phenotype. As you can see, Andy's a very handsome young man, but it has a striking psychiatric phenotype. And one of the most remarkable things is to go to a gathering of Fragile X families and see all the kids acting the same way, having the same little idiosyncrasies that you thought were unique to your kid. And it's really kind of freaky, but everybody is affected in a very similar way, just to greater or lesser degree. About 25 to 30% of males with the full mutation meet the criteria for autism, full-blown autism. All the rest have what we would now call an autism spectrum disorder, or what we used to call a pervasive developmental disorder. Small percentage have seizures. It's not like a lot of other rare epilepsies where people have 50 or 100 seizures a day. It's more like a couple a month. And so it's, by those standards, more of a nuisance than anything else. But when they're happening to your child, it's still pretty alarming. And Fragile X is a true developmental disorder. It's not neurodegenerative at all. So it makes a good model for the study of autism. It's an X-linked condition with highly variable penetrance. It's caused by an unstable trinucleotide repeat. And in fact, it was the first trinucleotide repeat expansion disorder. Huntington's is probably the most famous one, but Fragile X was the one that was discovered first. And the reason why it was discovered first is that it was named Fragile X because if you incubate chromosomes in a folate deficient medium, you can actually observe this fragile site in the X chromosome, so-called fragile site. It's not really fragile, it doesn't break, but it is distinctive, and it appears in about 20% of cells in full mutation males. And it turns out that that's right where the gene is, and that's right where this CGG expansion is. And so that helped scientists to locate this early on in the Human Genome Project. And so the gene was first identified in 1991, just before we got started, and right around the time our son got diagnosed. But this trinucleotide expansion leads to hypermethylation of the gene and silencing of the gene and loss of FMRP. Although it turns out in a new wrinkle that the gene may not be so completely silenced, but there's an effect on splicing of mRNAs. And so mutant splice variants of FMR1 get produced, but no protein gets produced. And the lower your levels of FMRP, the more severely affected you are. And importantly, we've had a knockout mouse since 1994, and it's a pretty good model for studying Fragile X. But what are we trying to do? What's FRAXA trying to do? What are you folks trying to do? Well, it's not basic science. This is an engineering project. We're engineering a solution for Fragile X. And that was something that was an observation that was made by Dan Vershbow, a grandparent, MIT grad, and an engineer, and a former board member of ours. May he rest in peace. Dan was an engineer, and he thought we needed to get some MIT people in on this to engineer a solution for Fragile X. And so that's the way we think of it. This isn't pure science. It's not pure clinical medicine either. It's more of an engineering project. We use what we jokingly refer to as the FRAXA method, which is we fund basic research. We've had a lot of basic research findings about the mechanisms of disease in Fragile X. That leads to translational research, which identifies treatment targets. When we have those treatment targets, we go ahead and try to treat them by doing preclinical validation studies of actual drugs, actual therapeutics. And if that's successful, hopefully that leads to clinical trials. And this has, in fact, led to many clinical trials in the Fragile X field over the years. Of course, this is not some proprietary method of ours. This is just rational drug discovery. And when rational drug discovery, which is what every pharma company in the world does, is done by a nonprofit, we think of it as community-based drug discovery. So a community of people with Fragile X or with SYNGAP attempting to tackle this problem head on, admittedly on a shoestring budget, on a nonprofit basis, in collaboration with academic labs to do a lot of this work, maybe contract research facilities to do the preclinical validation, and with pharma partners to do clinical trials. Perhaps the best example of this Was in the year 2000, we were funding a basic research project with Dr. Mark Baer at Brown University, an outstanding postdoc named Kim Huber. And they were studying synaptic plasticity in the Fragile X mouse using electrophysiology and pharmacology. They were studying a particular form of synaptic plasticity called metabotropic glutamate receptor long-term depression. And you don't really need to know the details about that, but just know that it's important. It's especially important for synaptic pruning and maturation of synapses. And because of the effects that the Fragile X protein were known to have in regulating activity-dependent protein synthesis, they had this process which was protein synthesis dependent, and their hypothesis going into the study was that they would see a deficit in mGlu-R LTD. So they were surprised, and in a bit of serendipity, they discovered that there was, in fact, excessive mGlu-R LTD. So there was too much of this form of synaptic plasticity in Fragile X, which has the net effect of keeping synapses in a relatively immature state. But what was really important and immediately obvious to everyone was that blocking this metabotropic glutamate receptor could help to rescue this particular phenotype. So it could fix this problem. And every major pharma company in the world was working on metabotropic glutamate receptor antagonists at this time for a variety of indications. So we started out with the preclinical validation by getting a research reagent, not necessarily a human drug, a little too toxic for that, but one that was originally developed as an mGlu-R5 antagonist drug called MPEP. We ordered some up from Tokris, and we organized preclinical validation studies in the mouse model with an old friend of ours who was a MD, PhD, and an expert in transgenic mice, Rob Bockwitz. And we showed right off the bat that you could treat the primary phenotype in the mouse model, audiogenic seizures, by treating the mice with MPEP. And in a nice dose response curve here, we see that the more MPEP you give, the fewer seizures you have. And in fact, these are perfectly normal, reasonable doses of MPEP that would be achievable in any animal and would correspond to human doses of an mGlu-R5 antagonist. And we could also show that the hyperactivity in the center square crossings in the open field would be reduced in a similar dose-dependent fashion in the knockout mouse with relatively little effect on the wild-type littermates. So this is proving out to be a fairly effective strategy. We did a lot more work than this, and I'm not going to kid you. It took millions of dollars worth of preclinical work to convince pharma companies that this was something worth investing in, but invest in it they did. And we eventually found a number of different pharma companies who were interested in Fragile X trials. There was a UK-based startup called Neuropharm that was working with an older compound called Phenibam, something that was off patent, but they were interested in developing it for Fragile X. They did some phase 1 trials, which showed it was very well tolerated, had some positive clinical effects that were observed, and it also rescued pre-pulse inhibition, which is not an approvable endpoint for the FDA, but it does give us an indication of target engagement. It tells us the drug is getting in there and it's doing something positive in Fragile X. Seaside Therapeutics licensed STX-107, which was a Merck compound, which is far more advanced than the older Phenibam. They did some phase 1 trials with normal volunteers that showed it was very well tolerated at extremely high doses. So it was something that you could certainly give to people. Novartis was convinced by the entry of these smaller guys to go with their far more advanced compound mavoglurant, which they had had in clinical trials for anxiety disorders. They did a phase 2 study, which showed a pretty robust response in a subgroup of Fragile X patients with full mutations, especially with what they called the fully methylated full mutations. So people who just weren't producing any of the protein at all. And of course, Roche, which is a mile down the road in Basel, felt compelled to jump in at that point with large-scale international trials. And so what happened? Well, Neuropharm went out of business because their lead compound, which was fluoxetine for autism, actually failed in its trials. Seaside Therapeutics went out of business because their lead compound, which was arbaclofen for autism and Fragile X, failed in its clinical trials. But of course, Novartis never runs out of money. And so they went ahead with very large scale international trials with more than 300 subjects enrolled. Very well done trials, but they came up with a negative result. The drug did not make people better. In fact, there was even some evidence that it resulted in a slight worsening and in a dose-dependent fashion. The higher dose groups did worse than anybody. And similar results at Roche, they were not as forthcoming with results as Novartis, but we've since come to see that their result was more or less exactly the same. So what happened here? Is this a problem with the method? I mean, is there a problem with rational drug discovery in Fragile X? Are we doing something wrong in some of these stages? Well, of course, the clinical people said the mouse model was no good. The lab people said, well, the outcome measures in clinical trials are no good. Everybody was blaming somebody else. But to make a long story short, what we saw in the clinical trial where our son was actually participating was clearly tolerance developing to the effect of the drug. And it's really important to understand that we validated a lot of other treatment targets. And so we want to make sure that this way of doing things is valid in and of itself. And, I think that we have a lot of other options in Fragile X, but if our methods for validating these treatment targets aren't themselves valid, well, we're just destined to have one trial failure after another. But in this case, it really was tolerance, and it turns out that there was research evidence for tolerance to the drug. And here you can see the tiny little fine print blown up from a paper that I was a co-author in 2002 that showed that, in fact, we did have tolerance to repeated doses of the research drug MPEP. There's some evidence that this is actually worse in Fragile X, if not actually unique in Fragile X compared to the normal population. So the pharma guys at Roche and Novartis and Merck and elsewhere all said that no, tolerance isn't going to be a problem. But it turns out that in Fragile X, it was a problem. Now, this is an iterative process, and this is one of the things you need to understand. We have this cycle here where we recognize that we have a problem, and we do the basic research to understand the mechanisms of disease. We do the translational research to identify the treatment targets. We do the preclinical validation where we test actual therapeutics. Then we take it into clinical trials, and it always looks good, or we wouldn't be doing the clinical trials. But the fact of the matter is, it doesn't always succeed. If you do succeed, great, you're done. You sail off into the sunset. But 90% of the time in brain disorders, clinical trials fail. And so then you have to spin this cycle again. You just have to understand that's the business of drug development in brain disorders in general. And I'm a psychiatrist and a psychopharmacologist, and I've seen any number of treatment strategies come and go over the years. But finally, we got a break, as one of these other targets was phosphodiesterase. And so we got our hands on a really good phosphodiesterase inhibitor that was in clinical trials for Alzheimer's disease. But it may actually be better applied to Fragile X. And we had a lot more support for that than anybody in the Alzheimer's field has. And so we tested this in our drug validation facility, which is, by the way, at the University of Chile, and with Dr. Patricia Cogram. And studies went very, very well. The study drug, BPN 14770, which is now known as zatolmilast, rescued all these various phenotypes in Fragile X, from open field to social interactions to nesting behavior to marble burying. So across the board, we got these really robust rescue effects at the doses that we would expect to use in humans, actually lower than the doses that were being used in the Alzheimer's trials. This was now being done as a 2-week administration of the drug, because remember, we'd been burned by the development of tolerance, and even 5 days of an mGlu5 drug would cause tolerance. So we studied this with 2 weeks of dosing twice a day. So we're exposing these mice to the drug for a long time, and we're getting a really nice effect, no tolerance. So then after exposing these mice to the drug for 2 weeks, we took them off the drug for 2 weeks and retested them. And this looks like I've made some sort of cut and paste error here, but in fact, this is a completely different set of experiments. And you can see that the drug works almost as well after the mice have been off the drug for 2 weeks. So we have a huge carryover effect, which to us indicates structural changes in the brain. So you're really doing something to change the wiring of the brain. And importantly, it translates. When we get to clinical trials, and we got to an initial phase 2 study in Fragile X with 30 subjects in a crossover design. So everybody got the drug, everybody got placebo. Some people got the drug first, then the placebo. Some people got the placebo first, and then the drug. And we saw across the board improvements in just about all of the assessments that were done. And in fact, a lot of the assessments aren't shown here because we also had improvement in human electrophysiology, in the EEG signature of Fragile X, and in auditory evoked potentials, which are excessive in Fragile X. So it dampened down excitatory sensory hypersensitivity. And so we got these across the board effects that were all positive, and some of these things were actually statistically significant. And these have become now the primary endpoints: the NIH Toolbox, Visual Analog Scale, and also some of the electrophysiologic measures in large-scale clinical trials that are now going on with Tetra and Shionogi. So we're enrolling, again, hundreds and hundreds of subjects around the world in large-scale clinical trials, and Shionogi is investing more than $50 million in this project. So that's what we like to think of as leverage. We have leveraged these investments in basic and translational research and gotten large-scale investments from pharma companies. People always ask me, because we've been at this for so long, Mike, if you had it to do over again, what would you do differently? And, the fact of the matter is we do have some regrets. We didn't do everything perfectly the first time around. One of the regrets we've had is that we've too narrowly focused for too long on specific aspects of Fragile X. It's the paradox of getting results. You do some basic research studies, and you assume that if you have a finding, well, that's it. That's the problem. But in this case, we identified that the Fragile X protein, FMRP, regulates protein synthesis in response to synaptic activity. But it turns out it does a lot more than just that. It also binds directly to ion channels to regulate excitatory neurotransmission directly. And it's not just postsynaptic, it's presynaptic too. So it's doing things in the dendrites, it's doing things in the axons, and it's not just neurons, it's also doing things in glial cells too. So FMRP is important to the function of astrocytes and microglia. And it turns out that's a very strong phenotype. And if you can just fix what's going on in the astrocytes, you can actually make a huge improvement in the overall Fragile X phenotype. But it's not just the brain, it's metabolism too. So I like to describe this as the streetlight effect, which is a well-known effect. It's a quirk in the human brain. It's something that's been studied in cognitive psychology. Starts out with a joke. A policeman is walking his or her beat late one night and comes across a fellow possibly inebriated on his hands and knees on the sidewalk, and he's obviously looking for something. Cop says, "What are you looking for?" "I'm trying to find my keys," says the drunk fellow. And then the policeman looks around, doesn't see anything, and says, "Are you sure this is where you lost your keys?" He said, "Well, no, actually, I think I lost them a couple of blocks back in a dark alleyway." And the policeman says, "Well, why are you looking for them here?" He says, "Well, the light is so much better." And this is what happens in all of science, unfortunately. In all of science, we have model systems and we have experimental methods. And that is the streetlight. And that's where we look. And there's often a lot of other stuff going on that we don't see as a result of that. So part of my role, at least I like to think, as medical director and CSO, is to try to shepherd the science and to make sure we're not overlooking something. We're not focusing too narrowly on any one thing. And we can do that by incentivizing other kinds of research. We also have come to appreciate that drug repositioning is harder than expected. Failure is often a prerequisite. In other words, pharma companies are not going to let you have access to their drugs unless they've already started running into a problem with that. The Tetra trial notwithstanding, that was the first PDE4 inhibitor of any quality that we could really get our hands on to do a clinical trial. And even that has run into trouble in the Alzheimer's studies. So the timing just worked out perfectly on that one. But there have been lots of other cases where the companies only allowed us access to their compounds or only became interested in a Fragile X indication after they ran into trouble with other programs that they had for that same compound. But drug repurposing is just the opposite. So you have drugs that are actual available compounds on the market. Now, repositioning is where you have an investigational drug that was developed for something else, and you're trying to use it for your rare disease. Repurposing involves available drugs that are already on the markets. They're already proven to work for something. And that usually means they're also quite safe. And that may even be necessary for rare disease foundations, because if you think about it, you need to develop the capacity to do clinical trials and to do clinical trials well. And how do you get good at doing clinical trials? By doing clinical trials. So you don't want your first clinical trial to be a trial of an ASO or a gene therapy product. You want to have the methodology worked out before you get to that point. And it's much easier to conduct clinical trials with available drugs. You don't have to ask the permission of the FDA. You don't have to modify your protocol. You can just do what you want to do with the available drugs, and you can fine-tune your trials methods. In addition, if you're using small molecules, you can imagine with any neurodevelopmental disorder, combinations are probably going to be required. I mean, we can't compensate for the complex functions of a protein like FMRP with one small molecule. We can partially offset the effects, but if we start to get into combination treatments, then we can do a whole lot more. Well, it's almost impossible to study combination trials with investigational drugs. You really have to start with available drugs to do that. And there are many ways you can do drug repurposing. One of the ways we've done is to use AI. AI is all in the news. Investors are all really excited about the potential of artificial intelligence. Well, you can actually use artificial intelligence to make drug predictions. And we actually did a couple of studies with a CRO called Helix. And they made a number of really interesting drug predictions for Fragile X, all done in the computer. One of them was for fenformin and metformin, which, unbeknownst to Helix, was actually being studied by 4 different labs that were funded by FRAXA, and subsequently was validated as a treatment strategy and is in large-scale clinical trials right now. So that gave us some confidence in their prediction methods. They also pointed us toward quercetin, which is an available nutraceutical, just over the counter, don't even need a prescription. Functions as an anti-inflammatory, antioxidant, and also has effects on AMP kinase, which is the same target as metformin. And so that's something that we've also been looking at, but also using clinically. I've got patients in the local area with Fragile X that I treat, and I've had some where I've supplemented their treatment with quercetin with great effects, and people really have been very happy with that. They pointed to some things which didn't work in preclinical validation, lest you think that everything works in the mouse model. Disulfiram, which is antabuse, didn't work at all. But we found that out by having the right tools to assess these compounds, and we were ready to go with that validation. And they also had some other predictions like Solindac, which is a non-steroidal anti-inflammatory drug, which was just about to go into clinical trials when the pandemic came along. And it's actually, frustratingly enough, it's still on hold.
Sorry to cut you off, Mike, but we're a little over time.
And so I'm gonna just wrap this up by saying that repurposing is really powerful. You can study things that are available compounds, like we've looked at things like caffeine and quercetin, I mentioned, in combination. But we've also informed our choices of medications by the things that we study in the animal models, and we know that certain drugs can replicate these effects. And so we can actually choose, for example, the best SSRI or the best atypical antipsychotic based on some of the research that we've done. And so this is a really powerful technique to assist in your clinical repurposing. So I'll wrap it up with that and just say that fortunately we've done a few things right, and I see that you guys are doing a few things right as well. And we wish you all the best of luck, and I'm happy to consult anytime with folks in the foundation. Best of luck to you, and there's my address, so you can send the swag. And I will stop sharing.
Thank you so much. So Mike, I really appreciate you taking the time to share that. I'm going to come into the mic. The reason I was so excited to have you talk, Mike, was so people could see everything you've been through, that you were excited, you had 4 trials. I mean, you've been at this for 30 years. And I just want people to be inspired by your progress and inspired that we can learn from everything you got right, but conscious that there's going to be fits and starts. And so we have we chronically overfill our agendas. So I'm getting snake eye from somebody right now. But as a closing thought, we are 5 years in. We are working on genetic therapies. We are working on repurposing. And you're talking to a room full of maybe 50 families. What would you want them to hear and think about as they're looking to the future and weighing their options between holding their breath for a genetic therapy, working on repurposed drugs, or working with clinicians to use existing therapies to treat symptoms?
Just remember, it's not one thing or the other. It's not either or. We think of it as a pyramid. So we have this pyramid, which the base of that is all of these repurposing studies looking at developing available drugs that we can just use right now or just make better choices. So the research can inform the routine, everyday clinical practice right now. But then higher up on the pyramid, we have targeted investigational drugs that actually go right to the heart of the problem and address the specific disease mechanisms you have. And then at the tippy top, you have at the top of the pyramid, you have these definitive therapies. You have the cure. And it's kind of a progression because all of these things reinforce one another. The repurposing studies reinforce the development of the investigational drugs. The clinical trials that you do with small molecules reinforces the capacity to validate ASOs and gene therapy protocols. We've even got studies of mRNA vaccine technology, but you have to have the animal models to work on. You have to have the cell-based systems to work on. You have to have the understanding of the disease mechanisms to do all of that. So realize that it's a comprehensive effort, that it does sometimes take a little bit of time, but you also get lucky along the way, and there is room for serendipity. But it's funny how the people who get lucky are the people who are working at things for a long, long time.
That's a perfect setup. All right. With that, Mike, thanks so much.